Welcome to the video on technical tips. I am your host, Dr. Monica Tyagi. I'm the application scientist with Biotechnic, and today I will be talking about tips and tricks to optimize your AAV analysis using our Maurice platform. The Maurice system enables protein identity, charge, size, and characterization applications in gene therapy for discovery through GMP product release. The Maurice system, it can do your icIEF and CESDS analysis. The MauriceFlex system, it can do both icIEF, CESDS, and additionally, it can fractionate your icIEF separated charged variants. Easy method transform from Maurice analytical icIEF to MauriceFlex icIEF-fractionation. Now, let's look at our latest system, MauriceFlex. Does for AAV. It can fractionate charge variants for downstream analysis with various methods. It is an offline CE-fractionation method offering flexibility for the type of downstream analysis you want to conduct. It could be mass spec, peptide mapping, or potency assays. The majority of current cell and gene therapies leverage two key viral vectors. They can be classified by where they are used. For in vivo delivery, LNPs and adeno-associated viruses are leading the way. For ex vivo work, cells are typically extracted from the patient and modified with lentivirals to provide the cells an advantage in finding the disease in the body. Now, let's talk about icIEF method development and flex fractionation of adeno-associated viruses. To begin with, your method development, we recommend our method development guide and app note as a good resource you can use using the default settings and the method provided, as these have been tested by our internal R&D and app team extensively. Once you get a good starting point, you can start working on developing your method further by keeping two critical points in mind. First is reproducibility, and second is resolution. As I mentioned previously, look for our method guide and app note. We have exclusive app notes on AAV for our icIEF method. Now let's take a look if we start developing a method using our default settings and the setup from the method development guide. And we are interested in developing an intact AAV icIEF method. What you see here, it's a hypothetical electropherogram. On your left, say you started with developing a method and you start using 4 molar urea as a solubilizer and a Pharmalite 3 to 10. What you notice that there is a lack of peak resolution. Now, how would you approach to resolve those peak initially? To improve the resolution, either you can increase the urea concentration or you can try different solubilizer. Example on the right, what you see that we're using simple salt. That's another solubilizer, and we see better resolution of peak. Try to change one parameter at a time so you can easily navigate into developing a good peak profile. For AAV, we recommend that you use fluorescence as a detection and at a higher exposure. Based on the peak profile, if you still want to achieve slight resolution, say, for example, your two acidic peaks are way closer to each other and you want them to resolve further, or your basic peaks are closer, how can you do that? You can check by using a narrow range amphylite. That selection will depend what's the PI of your protein is. You can work around checking the different concentration of these pharmalites. Additionally, you can also try by different concentration of the solubilizers. And then the combination of all of it can help you achieve the best profile. Also look into the IV plot and see how the separation looks and how the current looks. The narrow range amphylite in the abbot, I saw the profile that I showed you in our previous slide, it helped improve with the peak resolution using the simple sol method. Let me talk more about what can we do if we want to resolve those closely spaced acidic peaks. Here, you can see different amphylite combinations you can test. You can increase the final ampholyte percentage and adding narrow to wide range ampholyte. You can mix different manufacturer ampholyte. And this was just the example which I want to show that how many permutation and combinations that can be run to get and achieve what you're looking for. Are we ready to fractionate these peaks? These steps are somewhat similar as we recommended for our icIEF method development. We highly recommend that you look for our method guide for flex fractionation and the app notes. You can start with the default methods and the settings that are being mentioned in our method guide. When you start separating your protein and this charged species, look into the separation profile and the IV plot. Check for the mobilization IV plot, and it is very important how you prepare your sample for icIEF verification. I'm going to talk about these individual points in the forthcoming slides. I would like to highlight on this excellent resource that you can use for AAV capsid protein fractionation on MauriceFlex. This is from our App Science team in collaboration with Pfizer. Here is another helpful resource, a technical note, which can help you optimize your icIEF-fractionation method on the MauriceFlex system. The simple PI Western markers, which incorporate bright fluorescent signal on a plate reader, can help you reduce the guesswork in fraction identification and save time in fraction reanalysis. It's an easy method transfer from icIEF to icIEF fractionation. The only difference would be that we recommend using the higher concentration of your sample when you're planning to fractionate those charged species. For AAV, we would recommend go up to 5-fold higher the concentration what you have used for icIEFs. Additionally, we include 25 millimolar ammonium acetate that will help in mobilization of these fractions. It is important to look at the IV plot of your mobilization. The plot on the left corner is something you should be expecting. However, depending on the solubility of the fractions, you might notice dip in the current and lack of mobilization. In this case, you need to work on optimizing the mobilization and testing different mobilizers. In the webinar that I mentioned previously on denatured AAV, they tested different mobilizers to fractionate AAV, and that's an excellent resource. On the Compass software, this is how the mobilization will look when you choose fractions corresponding at different time. You can see the profile shifting toward the right end of the capillary, and finally the fractions getting eluted. You can change the mobilization and elution time setting based on your charged species. However, it is recommended that you start with the default settings as mentioned in the method development guide. Each molecule has its own set of challenges. In particular for AAV, it could be the low sample stability. In that case, have small aliquots and avoid repeated freeze-thaw of the sample. The current loss during mobilizations, probably due to the lack of solubility or low solubility of the fractions. Test different mobilizing solutions. Navigate the best sample concentration for good separation profile and signal intensity. And last but not the least, if you are proceeding for the CIEF verification step, first of all, for AAV, try using the low binding plate so that you don't lose your fractions. Avoid multiple pipetting steps. The more transferring steps are involved, the chances of losing those fractions would be more. Finally, you can run the icIEF verification from the fractions that you have collected from your flex fractionation. In conclusion, MauriceFlex support extended characterization and product monitoring, icIEF analysis of AAV for identity and stability indications, AAV capsid protein charge variants can be fractionated from downstream analysis, and easy method transfer from Maurice analytical CIEF to MauriceFlex fractionation. Thank you for watching this video and hope you find this video helpful in your method development.